Welcome back. In this third video, we want to think a little bit about extracellular matrix and how the cells might be migrating and try to piece it together a little bit. I keep forgetting to set my pointer, sorry. In the end of the previous recording, I just quickly mentioned this, my prediction that angiogenesis is most active in site 1 and not so much in site 2, simply, other than the appearance of the endothelial cells, just characteristic, the hypertrophy of them and the thickening of the basement membrane. But anyway, the appearance of the alpha-6 integrin staining being detected only in site 1 and really not at all in site 2. I think you'd have to agree that this is really nearly a complete negative stain. Hard to say if any of these little spots are things that would be considered truly positive, but not as distinctive as this for sure. And then based upon this dissertation that I found, the integrin A6, alpha 6, was associated with the angiogenesis, so that seems to be very supportive. So we start thinking about now, how can we have the migrating cells not get on blood vessels here? Are they getting on axons in site 1 is my prediction. I showed you the synaptophysin, and then migrating away on the axonal fiber tracts from site 1. And then on site 2, are they jumping on the blood vessels? So what's the route by which, and what is the route that they might be taking? Where can they spread in the brain? We see here in the rat brain, stroke or whatever might recruit them up there. So, which ones are moving, and how are they moving, and where are they moving, and how are they getting through the extracellular matrix and on, moving along other cells? Is the mechanism and everything the same? Here we see the illustration of the subventricular zone in humans versus that of mice or rats. Is it the same? Are these neural stem cells becoming brain tumors, or are they becoming other progenitor cells, then brain tumor stem cells? Can they be differentiating into different lineages? The glioblastoma is called anaplastic because felt that there's really no differentiation, but there might be partial types of phenotypes represented, and that's the stand that I take as we proceed, as you will see. There's even ideas, might be very theoretical, related to schizophrenia, but anyway, of many different routes that can be discovered by these progenitor cells, and it's all very consistent with what we saw originally of the spread of this. So how might that be occurring? How do we begin to take into account the local niche, the extracellular matrix contribution of the ependymal zone, or subventricular zone as it's called? This is the one in the hippocampus that we're not dealing with here. But we have the astrocytes, the neural stem cells, the transit amplifying precursor in green, the ependymal cell in yellow, the neuroblast in red, and we see it right on the blood vessel, and then of course the granule neuron is only for the hippocampus. So we extend basically those same cell types, and we start thinking of it here near the lateral ventricle, and we introduce this idea of fractones from this site, the glycoforum. And we start asking, well, what's the extracellular matrix in this area? These fractones, shown in the yellow, are laminin-rich structures continuous with the basal lamina of the blood vessels. So we've talked about blood vessels coming from the pial surface, getting all the way in to the ventricle, a long route, but that's how they get there. And how are these cells triggered and recruited to migrate away? The B cells here have astrocytic features and are considered the stem cells of the adult brain as far as replacing glial cells. The A cells are the proliferating neuroblasts, right, that migrate rostrally to the olfactory bulb, while type C are the stationary, very rapidly dividing cells. So some different characteristics. Maybe this type collection of cells en masse get represented in the glioblastoma tumor. Sorry for the delay there. And then over here we see the hyaluronidase has to be activated. We've got all of our different cells. So with the hyaluronidase activated, then these cells are freed up to migrate and move away. So these are all the things that we're trying to take into account in this. Sorry for the delays there. So we're thinking about what might be the preferences of which type of cell to stick to what. We see here the glial restricted precursor cell. It's has an initial lack of NG2 and platelet-derived growth factor receptor alpha expression, and their limited adhesion to laminin and preference for fibronectin. So we see the fibronectin staining over here by another group, only around the blood vessels. So if fibronectin is restricted to that, then is that the only place that we can see these cells migrate away? So is there a heterogeneity of the cell population and preference for extracellular matrix to bind to? So if we talk about the limited adhesion to laminin, how is that related to the fractones? So these are all questions that we want to think about when we're picturing the cells, making connections and walking away. And we're thinking about what type of fibers will be in the processes proximal to distal and at the cell body, and of course forming this nuclear cage is a term that I saw for it, and connecting to the centrosome. So there's a very complex mechanism for this migration. We see it here again. I just wanted to show this one to reveal some of the lists of the different markers that might be used to identify the motile cells that are creating these nuclear cages. And just to give some insights, are all of these the same or different depending upon the matrix that they're attaching to? Remember, we're thinking about axons or to the oligos lining the axons, or to the vasculature. Is it the basement membrane of the vasculature, directly on the endothelial cells, or just underneath the astrocytes? What really is being involved? One or two or more, or all of them? So we are picturing the migration now away from the primary site, and you can see the text here. In vivo, the neural stem cells can track invasive glioma cells migrating from the main tumor. So there's more and more information becoming available, but to bring it back to what we're interested in, the nestin-positive cells are invading the normal tissue. So here we see the tumor, similar to what we have here, and the nestin staining is revealing that. So our nestin staining is very likely to be associated with the migrating cells. This one is a very nice study in 2011 in Cancer Research. It's a 3D reconstruction of the tumor itself and the migrating cells, and then the vasculature. And here you can see the migrating cells very nicely jumping on the vasculature to move away, I guess, migrate away from the primary tumor, because these are implants in the rats, of course. So we are always continuously thinking about what's the source, transformation into the tumor. Can it go back and forth within the tumor? Are we getting any representation of these types of cells? Here we see an oligo in red and the astrocytes in green. We get a cross section through a brain tissue, even though this isn't culture. What would those look like? Here's the astrocyte and neurons. Going to see more fibers than we are cells. So the histological stains that we look at, in particular the vimentin, that's the point I keep making about these, which types of these fibers, the oligos, the astrocytes, or are there neurites in there that are going to contain the vimentin. And again, we're always thinking that we could have any of these cell types represented in the tumor. It could be differentiating into ependymal cells. They have to be replaced, so the stem cells should be replacing those. We see the vasculature, the astrocytes, the microglia, the neurites, and the oligos. So anytime we look at a cross section through a tumor, which of these might be represented in there? And again, just reminding ourselves of all the potential interactions and the molecules that might be contributing to all the events that are occurring in the tumor itself. So we'll stop there for this recording, and we'll pick up with this for the next one.